Hello and welcome to our RX Hope Talks, Table Talks, Session 11, the secrets that animal models reveal about SCN2A. This session is being recorded. My name is Catalina Betancourt, and I am a board member of the Family SCN2A Foundation. We are very excited to be bringing this virtual education experience to our global community. During these sessions, the family and caregivers of the patients with SCN2A related disorders will be referred to as the experts, and the presenters will be the professionals. The goal of the virtual table talks is to allow experts, meaning us, access to professional researchers and clinicians working diligently to find a cure for our children. This is meant to be a casual environment, and your questions and comments are encouraged, as well as we are all here to learn from one another. But to keep things running smoothly, please keep your audio on mute and use the reaction button to raise your hand if you would like to talk. Also, you can send questions through the chat. If you haven't already, please introduce yourself and your SCN2A child in the chat. You can perhaps say their name, age, and how SCN2A presents in them, and where you live. I now have the pleasure to hand this over to a longtime friend and advisor for the foundation, Dr. Kevin Bender and Dr. Jennifer Kearney.
Thank you. So our title is the secrets that animal models reveal about SCN2A. And for full disclosure, I'm not sure how many secrets we're going to reveal, but certainly, hopefully by the end, you'll understand maybe why we're looking at different animal models and what we hope to learn from them. So over the course of this seminar series, you've probably heard about a variety of model systems, some of which are illustrated here. And each system is valuable for addressing different types of questions. However, these often exist in silos. So there might be an iPSC lab or a heterologous cell model lab, and without a lot of crosstalk in between them. So one of the major questions we've been trying to address in our channelopathy-associated epilepsy center without walls is, what are the salient features of each of these model systems if we look at the same variants across them? And can we use that to understand disease mechanisms and hopefully make predictions about what treatments might work best given a particular variant? So my charge within the center is to make mouse models and to characterize the in vivo phenotypes as well as pharmacology. So today what I wanted to do is just talk a little bit about how we actually make the mouse models and some of the lessons that we've learned along the way in the process of making them. But first, I wanted to step back a little bit and just talk about why we need animal models to begin with. And although I'm going to speak specifically about mice, Kevin's going to talk about some other animal models that might be useful for addressing some phenotypes where mice fall short. So again, the animal model that you use really depends on the question that you're asking. But the reason for using an animal model is that they provide access to a developed or developing brain. So I'm kind of a simple person. I let the mouse make a brain for me instead of trying to figure out how to make iPSCs into a brain-like organoid. We can also use them to study impact of pathogenic variants on fundamental biology, so kind of very basic questions about mechanisms. And we can do that at multiple levels of analysis when we have an animal with a brain. So we can look at the level of neurons, synapses, circuits, networks, systems, so maybe not just the brain, maybe we want to look at some of these other systems in the body that might be affected. We can also use the mice or other animals to identify biomarkers of disease progression or treatment response. One thing that we do a lot in my lab is look for genetic and environmental modifiers that might impact the phenotype and could explain why two kids with the same exact variant don't look the same. We can also try to provide additional evidence or identify novel therapeutic targets. And the animal models can really help prioritize candidate therapeutics for future clinical trials. So if you have multiple analogs of a specific novel compound, you might make a decision on which one to take forward based on some of the animal studies. And finally, one of the really important things from the standpoint of the FDA is evaluating safety of novel therapeutics. So usually you need safety data from 2 different species to move forward if it's never been in people before. So those are perhaps not all of the reasons, but a lot of the reasons that we use animal models. So how do we make them? So we've been using CRISPR Cas9 editing since 2014, when I think 2013 was the first CRISPR edited mouse, and it really has been a game changer. It's completely changed the way that we make mouse models, and it's changed the time scale. So we can do something in 3 or 4 months that used to take 12 to 18 months, and it's about 1/5 of the cost, so we can make more models. So it's very efficient. And one of the things that we've really learned along the way is it's almost too efficient for genes that have homozygous lethality like SCN1A. So just as an overview, what we usually do, you take Cas9 protein, pair it up with a guide RNA that matches the sequence that you're trying to target, and you assemble this in vitro into a ribonucleic protein complex. And if you're targeting for a knock-in, there's also a template strand that will introduce that variant. And this generally gets injected into fertilized oocytes at the single cell stage, which are then implanted into females, and then you get your mice out the other end. Because you're injecting into a single cell oocyte, basically all the cells in the body are going to be targeted. And because it's so efficient, CRISPR almost always targets both copies of the gene that you're trying to target. And for SCN2A, we know that we need at least one functional copy to keep the animals alive. So we did a lot of rounds of targeting with our transgenic core and ended up with great pups are born, and then the next day there were no pups left to genotype. So we really needed to step back, and I met with the transgenic core people quite a few times and said, can we make the process less efficient? Their goal is always to make the process as efficient as possible. And so then what we decided to do was step back and say, what if we inject at the 2-cell stage instead of at the 1-cell stage? And in that way, half the cells would be targeted, half of the cells that make up the animal would not be targeted, and we end up with a mosaic animal. So some cells have that targeting event, some don't. And what this allows us to do is actually get animals that are alive that we can then breed, and then we can generate heterozygotes because we just breed them to wild type mice, and those will survive, and they model those heterozygous variants that are in patients. So probably the question that you all think about a lot is, how do we pick which variants to make mouse models for? And there are a number of criteria that we use. One of the most important is it has to be tractable for CRISPR-Cas9 editing. It requires a specific sequence tag. And there are other editing systems that we could use, but CRISPR-Cas9 is sort of tried and true and is kind of the easy one to go forward with. The other thing that's super important for SCN2A is we have to find guide RNAs that don't hit other sodium channel genes that are in this cluster on chromosome 2. So these all arose from a single sodium channel, and there is a tandem duplication. So they have very high sequence identity at the nucleotide level. So between SCN2A and SCN3A, there's about 85% nucleotide sequence identity. And for SCN2A and 9A, it's about 83%. So what this means is there are some portions of SCN2A that just are not accessible, at least by CRISPR-Cas9 editing, because we can't target just 2A. We will target 2A, and because it's so efficient, we will also target 3A and possibly 1A and 9A.
And Jennifer, that's what you mean, sorry, that's what you mean by tractable?
Yep. Okay. So they have to be tractable. And, you know, we definitely early days found this out kind of the hard way, and targeted 2A and 3A. And then because they're so close together, you really can't breed them apart. So if they were on different chromosomes, it wouldn't be as much of a problem, but because they're right next to each other, it's really impossible to get away from it. So there is still some place for homologous recombination, which was the old way that we used to make knock-in mice, but we can also just kind of pick and choose our variants based on that. The other thing that we look at next is we want to represent the major categories of channel dysfunction. So the way channels generally work is when they're open, ions go through them. And if the channel has a mutation that affects the permeation pathway, you might have a change in how or which ions go through the channel. So that's an altered permeation path. You can have altered gating. So the channels activate and inactivate in response to changes in membrane voltage. That's why they're voltage gated. And so altered gating is another property that we can look at. And then of course, loss of function or premature termination codons are another major category. And then one that I think has been particularly important to me is to make unique variants that haven't been modeled by other labs. I think with limited resources, we want to make sure that we're getting a good breadth of models, not having 5 labs making the same variant. And so after we went through those criteria, plus we have a panel of individuals in our center that by committee helps decide which variants get modeled. We pursued these 2 variants, so K1422E, which is an altered permeation path variant, E430A, which is altered gating. And then as a byproduct of this E430A, we got a frameshift mutation nearby, which is handy to have as well. So the K1422E mouse model is one that we've had for a couple of years. And I think I've talked about it to the SCN2A family group a couple of times, but things that we know. So we use CRISPR-Cas9 and the homozygotes die within a few hours of birth. For us, this is kind of a quick check of, did we target SCN2A? If the homozygotes survive, then we know we didn't do quite what we thought we did. The heterozygotes are viable and fertile. We can breed them. We can make as many mice as we want, and that's a good thing. We spend a lot of time going through and sequencing. This is just a sequence chromatogram that shows the codon 1422 and shows here that we have a heterozygous, so 2 peaks on top of each other, nucleotide at this position, and that's the K1422E variant. We also sequence the rest of the gene. We sequence other genes that are nearby, and we also sequence predicted off-target sites. So we do a lot of quality control at this stage before we get into phenotyping the mice, because we want to make sure we're actually looking at the right thing. They have normal amount of SCN2A transcript, normal amount of SCN2A protein. So again, important quality checks that when we made this mutation, we didn't somehow disrupt the protein expression. And so things that we do, I come from an epilepsy background. So EEG monitoring is something that we do routinely. And I'm going to start this video playing now, and it will play while I describe this EEG. So just kind of bear in mind this video is playing. So this is an example of a localized seizure. So A is a 2-channel EEG, and then this purple bar here where the seizure is blown up in B. So you can see there's an evolution of these high frequency, high amplitude spiking. And then we get slower spiking towards the end. This second channel doesn't have anything happening in it. So this is a localized seizure. It's staying within, so the channel actually runs from left to right across kind of the back part of the cortex, of visual cortex essentially. And so that's interesting. Now, this video has still been playing, and what you can see is that the mouse didn't do anything. So there's no behavioral components, which is also consistent with a localized seizure that doesn't go into motor cortex. So this EEG is lovely. The lack of a behavioral component means that it's very difficult to phenotype seizures unless we do EEG. So another thing we can do to try to make that a little more tractable is look at seizure susceptibility. So in this case, we use a GABA inhibitor called fluorothil, and it's delivered through inhalation. So the animals go into a chamber and they breathe in this fluorothil, and eventually all mice will have a seizure. And it's just a matter of when that happens. And what we see is that there's altered seizure susceptibility between the wild type animals and the KE animals. And these are males. So interestingly, when we do the same thing with females, we see something a little bit different. The average is still different, but we have two populations. We have one way up here with long latency and another down here with short latency. And this is one of those environmental modifier effects that we're interested in. We suspect that this has to do with circulating hormones, because these are adult animals when we're doing this. So we think that that might be playing a role, and we're trying to pursue that now. Another thing that we look at is strain dependence. So everything I've shown you so far has been on C57 black 6J mice, which are kind of standard mouse strains. And we can breed them with other inbred mice and look at what happens to phenotypes. And in this case, this is percent lethality from that fluorothil induction. So if we had about 30% lethality in the KE mice on black 6, and that goes to 0 when we cross to DBA. And conversely, the black 6, we have no lethality for black 6, and in the F1s with DBA, we start to pick up some lethality. So there's some strain dependence here, and that's something that we're interested in following up and trying to find the genes that are behind that. So we also did behavioral phenotyping. I'll just show you one assay for this. So this is the 3-chamber test that you've probably all heard about at some point, but it's a 2-phase test. In the first, we look at sociability. So the animal gets a choice between interacting with a mouse that is a new friend or an empty wire cage. And both the wild type mice and the KE mice prefer the novel mouse, but the magnitude of that preference is greater in the KE mice. So they have enhanced sociability. In the second phase, this same mouse remains. So now this is a familiar mouse, and the animal, the test animal here, gets access to a new friend again. So this is the novel mouse. And again, both the wild type and the KE mice prefer the novel mouse. So that's more interesting. But the KE mice have a stronger preference for the novel mouse. So again, enhanced social novelty. And in some ways, this is counterintuitive. You would think maybe it should be the other way around, but this is something that's been seen with some with this assay and some other SCN2A knockout and hypomorphic lines. And I think we can hand wave about what we think it means, but I think anything that's an alteration in normal behavior seems to be something that we can at least attribute to the genotype. So I would definitely say the most important thing that my lab does with mouse models that we develop is we disseminate them. So we disseminate the information in publications. So this is a bioRxiv preprint. And I don't expect you to be able to read this, but one of the things that we did is we sent these mice off on a journey out to San Francisco. And so down here is Kevin's name, and he can do all kinds of fancy things with the mice that we don't do.
The coats were a really nice addition. I appreciate that you send them with the scarves and everything.
Absolutely, they need to be dressed up and ready to go.
It is cold here in the summer.
But definitely, you know, there's so many things that can be done with different mouse models, and one lab can't possibly do them all. So that's definitely, I think, the most important thing that we do. And we've also started the process of depositing these in the Mouse Mutant Regional Resource Center, which is the NIH sponsored repository for genetically modified mice. The process is a little bit slower than I would like, but they had some COVID limitations, so hopefully they'll get in there soon. And the advantage there is then people don't have to contact me and I have to send them the mice. They can just go to this catalog, order the mice, and do their experiments. And that makes it so that it's democratized. People can do whatever they want with them, and it just makes it a lot easier in terms of animal care and not having to do quarantines and all kinds of things that happen when individual researchers ship mice to other individual researchers. So I have very little to say about the other two mouse models that I mentioned. So E430A is a relatively new model for us. We have confirmed that we have changed the glutamic acid to alanine here. We also had to make a silent change to disrupt the PAM site, but the amino acid sequence is the same. With these, again, the homozygotes die within a few hours of birth, so that's our checkpoint. And the heterozygotes are viable and fertile, and they have spontaneous tonic-clonic seizures. We can play that video. The end of one here, and it's on a loop. But that was encouraging because this was just in the course of normal colony maintenance, you know, going in and breeding animals and moving them around. So, and it's been seen more than once. So we think the seizure frequency is probably maybe a bit higher than the K1422E model. So the Frameshift model, aside from confirming that we do indeed have a deletion here that causes a frameshift, we don't know much about them. We predict that this will result in no protein, which would be equivalent to a knockout, or possibly low protein, which would be a hypomorph. And I think the Western blots are probably incubating as we speak. But the reason that we want to have a frameshift in our hands is so that we can actually do comparisons of the three different models side by side, because it's, you know, we can always compare to what other people have published or reported, but if the animals aren't on the same exact genetic background and aren't growing up in the same mouse room, they may behave differently. So really doing a side-by-side comparison within a single lab and really with a single experimenter can make a big difference. So just kind of the state of play of the SCN2A mouse models, at least as far as I know about them, and you all might actually know more. But as far as I know, there are multiple knockouts and conditional knockouts and knock-ins. And most of those are available through this international mouse strain resource, or at least a good number of them. So that's good. There are a couple of hypomorphic alleles that have been reported recently from Yang Yang's lab and Jeff Pitt's lab. I told you about the 3 that we've developed. And then these are at least 4 missense mutations that other labs have developed that I've heard about, haven't necessarily seen publications about them except one of them, and don't know the status of sharing. But I think really the goal is to get all of these models and more into these repositories so that they can be shared widely. And we get more information that way than when we keep them siloed. And I think that's where I end, and Kevin's going to jump in.
Yeah, so I was going to just show a few slides on some other model organisms that are sort of in development in the SCN2A world. And in particular, for whatever reason, we have a bunch of labs here at UCSF that have made inroads into SCN2A through these other model organisms. So the ones in particular that we've been working on in collaboration with 2 labs here are in rats and voles. These guys end up being somewhat useful for different things than mice. So mice have this wonderful ability of breeding extremely quickly, but they're kind of tiny, and there's only so much you can do with them, and there's only so much you can teach them. Rats, by contrast, you see this little rat, I think his name is Wilson. These guys have been named after presidents as they go. You see this little rat on the left side. He's wearing this giant hat connected to an HDMI cable. That helmet that he's wearing actually contains recording electrodes, and this is from Lauren Frank's lab just down the hall from me. And they are a lab that is built, and actually they designed these hats to do massive recordings from large parts of the brain, including the hippocampus, which is involved in short-term memory and learning your way around the world, navigation, as well as places that we've studied before in relationship to SCN2A, including prefrontal cortex and other parts of neocortex. And so we're just starting a collaboration with them to try to understand or see whether or not we can leverage rats as another potential tool to understand SCN2A. They're also, I would say, much more social than mice. Mice can kind of devil babies, I would say is the right scientific term, right, Jennifer? So rats are cute, they're cuddly. You can buy these same rats, they're called fancy rats. If you go to Petco, you can actually buy them. You probably catch them in your backyard if you don't want to buy them, depending on what part of the world you're in. Anyway, the other animal that's being developed nearby are SCN2A prairie voles. And this is work that's being done by Devin Manoli's lab. He's very, very interested in understanding social interaction and maternal behavior, paternal behavior. Voles have this really unique property of, amongst all of the rodent organisms that live in the world, they're pretty much the only ones that will pair bond with a mate for life, whether it be male female. Sometimes you get male male or female female interactions, but they'll find a mate and or a buddy, and they will be buddies for the rest of their life. And they're also extremely useful for understanding maternal behavior, nesting, basically huddling all their babies around. And so have been developed as this model organism to just try and understand a lot of the social aspects that are associated with things that we're generally trying to understand in the loss of function realm with SCN2A and its relationship with autism spectrum disorder and the social interaction issues that we tend to encounter there. And so these are 2 things that one could use. Eventually, one would imagine that there might be some non-human primate, and by that I mean monkey, SCN2A animals in the world. We have friends, both Jennifer and I, and we're trying to convince them that these model organisms should be developed, but they're still down the road. And then if you go in the opposite direction, you could work to something like a zebrafish. Those are a little bit more complicated because of the gross excess of gene copies that are found in zebrafish. And so it gets really complicated really quickly. And they generally don't have the same structures within their brain that mammals do, including us. And so at least at UCSF, we've been sticking with rodents of various shapes and sizes. So I want to show you one thing real quick that we can do in rats. And by we, I mean Lauren's lab. So that little hat, and Jennifer, you can go to the next slide. That little hat has a bunch of recording electrodes, and you can record from hippocampus. And what you see on the left are a bunch of tick marks in a bunch of different colors. Those are when an individual neuron in the hippocampus fires an action potential in that cell. And so each cell by cell number from 0 all the way past 45, those are all the cells that are active during this thing. And then on the right, you see an M. That's a maze. And this little green thing that you see right there is circled over the rat's head. And what you're going to see as the rat moves around is that there's a lot of activity in different parts of the brain or in different cells within the hippocampus as this rat navigates. So hopefully this will play. Yeah, and if you turn your volume up, you might actually even be able to hear some tick, tick, ticking. But you can see this animal is navigating from one part of a maze, the center arm of an M, all the way to another part of the M. And you can see on the left, there's this beautiful array of the activity because each of those cells are involved with encoding a particular place in the world. And so one of the big questions we have, and we actually have SCN2A heterozygote animals, rats in Lauren's lab right now with this exact same setup in their head, is asking the question, how well do they navigate these mazes? How well is information encoded and represented within the brain in these animals? And are there any changes in the way these animals learn to navigate these mazes? Because what this animal has to do is learn, okay, if I went to the center arm, I need to now go to the opposite arm and then go back to the center arm and then go back to this. And if I want to get my peanut butter, I got to do this. Turns out that SCN2A rats actually have a little difficulty with doing that. And that is something that we would never have been able to tell from a mouse simply because we can't train a normal mouse to do this complex behavior. This is actually too complicated for a mouse. And so we rely on rats, which can also carry a bunch more information. Okay, so that's the thing that we're seeing so far in rats. In voles, it gets even more crazy. I mentioned before that voles like to pair bond throughout their life. And so you can measure this by spending time huddling with your buddy. And so normally you'll just huddle with your buddy all the time. And this is work done by Gina and Narissa in Dev's lab. And if you introduce a novel animal to that huddle partner, a normal animal, which is called WT or wild type, they'll reject that animal. They'll actually kind of fight them and say, stay on the other side of the room. I don't want anything to do. I miss my girlfriend or my boyfriend. You stay away. And so you can see the lines for time huddling for the wild types are near 0 for both wild type females and males. The SCN2A heterozygotes, which are called D3/+ here, the males, I always joke that they turn into Republican congressmen from Florida. They're very, very promiscuous. I know that's a terrible joke, but I'm going to say it anyway because there's only 12 of us here and it's not being recorded. It's okay. So these animals, for some reason, either cannot remember or are just much more socially interested in the world. And this actually looks in some ways a lot like what Jennifer was telling us in the K1422E mice, where there was a good hint that something was happening, but it's certainly, I would say honestly, it certainly wasn't as robust as this effect that we're seeing here, which is clear as day. This animal really can't tell that this is my partner or not, or doesn't care. The latter would be more of a Florida phenotype, I guess. So that's an interesting thing, and trying to understand why that is or what is going on there, I think is also a very interesting thing to study. This, in some ways, I don't know if I could anthropomorphize this, but I've always noticed when we're talking with you and the family group and just meeting your kids, how cuddly they are. And are they cuddly with everybody, or are they cuddly with caregivers? I think that they're more cuddly than your part and parcel ASD kid who would like to kind of be on his own. And so maybe that's part of the phenotype, and we're starting to see that in voles. And I think, was there one more slide? Yeah. The other thing I wanted to show you is that we've been doing physiology across all of these different model organisms. And one of the things that's kind of exciting, and we also saw this in the K1422E animals, is that the phenotypes that we first identified in the mice in terms of how action potentials are transmitted in the cells are also present in the rats and are also present in the prairie voles. And that is actually a really beautiful thing to see. We've actually seen now, we've done work in borrowing some SCN2A heterozygote stem cells from humans from Jen Pan's group at the Broad. We've seen the same phenotypes in those cells as well, where basically everything has been consistent across every model organism we've ever looked at, which really lends to some credibility that what we're studying is something very central to the function of SCN2A. And so we're probably on the right track. So that's pretty much all I wanted to show today, I think. And we might have a conclusion slide. We do not have a conclusion slide. We're stopping there. Thank everybody in the lab who's done this. This is a lot of work from a bunch of different labs here at UCSF, and probably too many to name. So we'll stop at this point. And if you guys have questions, we can chat about some questions. And the kids are serial huggers. That is true.
Thank you so much, Jennifer and Kevin. I do have every single therapist that has seen my kid. She has, or he has, to comment always on how cuddly my son is. Sometimes he will see the therapist sitting somewhere and he will just walk towards the person and just like stay on that person. So I can't believe that. It's funny that you said that.
Yeah.
Yeah.
So the real trick is we've got to come up with a therapy that rescues everything except for the cuddling.
Yes, we want to keep that.
Yeah.
Yeah.
That's definitely a benefit.
Please feel free to send your questions in the chat or unmute yourself and raise your hand if you have any questions.
Yeah, I just saw a comment from Kevin about rabbits. And so for Dravet syndrome, there are rabbits. And that's super important for studying cardiac phenotypes, because this is another example where mouse physiology or cardiac physiology is extremely different from human. And so it's really not a great model for studying cardiac phenotypes, whereas rabbit is a lot more similar. So there is in fact a rabbit SUDEP model with an SCN1A variant. And
oh wow, I didn't know about that. That's great.
So that's really been the thing about CRISPR, is you can really use CRISPR on almost any model system that you can think of. I think zebrafish have actually probably been one of the hardest ones to get CRISPR to work. Whereas the old school way of making knock-ins and knockouts was really limited to mice for the most part. And I think rats were kind of just coming along, but were a lot more difficult. But with CRISPR, you really can make any kind of non-human primate model, sure, no problem. So that's really opened up a lot of possibilities, but I think we have to think about what questions you want to ask and what the benefits are. Because with non-human primate models, for example, you can't make a lot of them. And so you really need to know exactly what you're going to do with them, and that it's going to be worth the time and considerable expense of making them.
Yeah, I totally agree with that. It's a tricky thing to sort that out. There are cases, and I mean, we're getting to the point where SCN2A might not be one of those cases where it's very hard to study loss of function heterozygosity, which is what the human condition in many of these autism spectrum disorder genes, in a mouse. And one of the real good examples of that is a gene called SHANK3, where if you look at an animal that is heterozygous for SHANK, which should be a mimic of the human, it actually has absolutely no behavioral phenotype in anything. And you have to make a full knockout to actually start to see effects. Whereas when the macaque monkey version of that SCN2 SHANK animal was made, it had a clear as day social interaction deficit. That was very, very clear. So there is a utility for monkeys, but if you have the opportunity to work in rat models, mouse models, then it's much more amenable to doing the exhaustive everything from channels to behavior that many of these labs are doing.
We have one question here in the chat. How do you choose SCN2A variants to create a mouse model with?
Yeah, so the things that we think about in my lab is first, there has to be a sequence available. So there has to be a recognition site for the CRISPR gene that's within about 20 base pairs of what you want to target. So the SCN2A gene is quite large. There are regions where there won't be a recognition site near that particular place you want to target. And that's where you can go to alternative CRISPR. There are different Cas versions, and you can go to different Cas versions and maybe pick that up, or you can go back to homologous recombination. But really the most critical thing in our hands has been that similarity between SCN1A, 2A, and 3A. And there are just parts of the gene that just are not going to be targetable because you can't target 2A without hitting 3A and often 1A as well. So they have, 100% sequence identity over hundreds of base pairs. And I think we started with 7 nominated variants from our Center Without Walls committee.
Mm-hmm.
And at least 4 of them were eliminated based on that sequence similarity issue. And what it really comes down to, the parts of the channel that are really important for critical function are the ones that don't vary between the different SCNs. And those are also where mutations tend to pile up. So, evolutionary conservation is one of the things we use for clinical genetics to give, that's one piece of evidence. If something has very strong evolutionary conservation, that tells you it's a really important part of the protein. Therefore, you probably can't change it to anything you want and still maintain function. And that's also true when you have this fairly recent tandem duplication of these genes. So they were all one gene. And then in evolutionary history, it's recent. And then it expanded out into these 4 genes. So they really are the 85% nucleotide level identity is highly unusual. That's way more sequence identity than most genes. And that's just because it's a recent change. So they were the same thing, and now they've basically Xeroxed each other, and each copy gets a little bit, maybe different. You might have a dust speck here, and but essentially they are all the same gene at one point in time. So that's really a limiting factor. And then, it's been, I think a lot of people have wanted to go after recurrent variants. And that's been true for other groups in the world who have made like our A53Q and our 1882Q. Those are recurrent variants. It makes a lot of sense to make them because there are more individuals that have those variants. But to me, if I know another lab in the world that does good work already made that mouse, why would we make it two? I think we've kind of steered into let's make different variants, and if that other lab wants our variant, we're happy to send it to them. So, does that answer your question, Leah?
Yeah, it does. Thank you so much.
Okay.
I have a question. Oh, sorry, I'll just ask. How close are mouse models to people when testing drugs safely?
Yeah, so I think when we're talking about seizures, we're in a pretty good place. So seizures in rodents are similar, it's the same symptom as a seizure in the human. And there's a very good translational track record from mouse or rat models of epilepsy to drugs that work in people. So that's one of the successes in neurology of preclinical to clinical translation. Whereas you probably heard a lot that things that work in mice never work in people. And often that's when you're measuring something in the mice that you think might approximate what is a symptom in people. So like if we use that 3-chamber interaction test and say, that's going to do something for treating social behavior issues, we might not hit the mark on that because we don't know if what we're measuring in that assay actually has anything to do with behavior in kids. So I think we're in kind of a good place for the epilepsy portion of it. The tricky part is that in a lot of cases we're talking about pharmacoresistant seizures. So it's really hard to actually tell how predictive your model is when there's not a drug that works really well. So that's kind of, it's a chicken and egg problem. You kind of need a drug that works well in order to prove that the model has predictive validity, but you probably made the model because you need drugs that actually are going to move the needle in the clinic. So it's a little bit tricky. The other thing, at least with K1422E that we've come up against, is the seizure frequency is just extraordinarily low. So it's a little bit challenging when they only have a couple of seizures a month. To do in a month in a mouse life is a pretty long time. So if you only have 2 seizures in a month, that's kind of a long, drawn out study that would require probably hundreds of animals to see an effect. So that's why we do things like induce seizures using chlorophyll and other agents, but it's a trade-off.
So Michelle's asking on the chat, how can the foundation encourage researchers to share their mice? Is there a repository we should ask them to add to? Jennifer, you're the model citizen for that.
Yeah, so, you know, with our Dravet mice, we have shared them with, it's about 30 groups across the world at this point that I know of. We also deposit them in that Mouse Mutant Regional Resource Center. This one was at the Jackson Lab. We did that probably more than 5 years ago, and we don't know anything about what gets ordered out of there. So I do meet people who have ordered the mice from there. So I think the impact that the Dravet mice have made on, actually getting things, generating preclinical data and getting things into clinical trials in humans, like that's actually been a real impact of that mouse model. So sharing it with groups that are doing that kind of work really has made a huge difference. So that's, I think sharing is super important. The regional resource centers are great, except they freeze everything down. So sometimes it's faster for us to ship live mice.
Mm-hmm.
I think we set a record with shipping mice to Kevin, because that was like in the middle of COVID that the mice actually went out the door. It was pretty fast, all things considered.
It really was.
But if you had to get those out of a repository where things are frozen because they can't possibly keep everything live.
Yeah, I'm running into that problem. I'm running into that problem right now. We ordered mice and they're on a 25-week back order because of staffing at COVID.
Mm-hmm.
For a different project. It's not SCN2A. So we'll get them eventually.
Yeah, and if they're frozen, it takes a good amount of time, and it also costs a decent amount of money.
Yeah.
Versus getting the live mice it's not, not inexpensive. There are still costs associated with that, but it's definitely gets you doing experiments a lot faster.
Jennifer, have you tested with any of the models, have you tested any seizure medications? And then probably to spin off of one of the other earlier questions, and if so, do you take like data from that particular patient or from to make sure that the presentation of the mice is actually similar to the patient?
So we have administered drugs to the animals. I don't think that we have really systematically evaluated them, or at least analyzed the data that was collected, because we did a lot of it using EEG, and we're going to use background EEG as our signal, because the seizures themselves are not frequent enough, but background EEG can also give you a lot of information. That has not been fully analyzed, so I can't really comment, but absolutely knowing what drugs work and don't more often don't work in children with that particular variant is really important to try to understand whether or not it's a good pharmacological model. And even though we didn't pick variants that we were going to make mice of knowing who the kids and families were, we know who they are now, and you all know too. And so sharing that information, but also we don't want to try to force fit phenotypes in the mice to things that happen in kids as well, because they're we just kind of want to see what we see and then put those two things together. Because I think there's definitely a danger of trying to force fit phenotypes. If you're doing it ahead of time, I think it definitely introduces a bias
But just for families to know, because we're always encouraging to participate in research, and it takes a lot of time for families to participate and do the surveys and get their medical records and things like that. But is that a factor for you guys when creating a mice? If you're like between a couple different variants, but there's more information readily available from one variant versus another, then that might be your deciding factor. Is that?
So that actually is, so we have basically a panel from our center without walls, one of which is a clinical, so we have a clinical core that does all of the curation of patient variants and phenotype collection, drug response data. And so they definitely have a voice in the process. And I think, they always want to do recurrent variants. So that's really the only place where we've maybe not totally agreed. But yes, if we have a variant that we've found in ClinVar that just has, a variant of unknown significance and a 4-word diagnosis associated with it, we wouldn't make that. We need to know that there are real records and it's a bona fide pathogenic variant that the clinical geneticists in the group have agreed is actually likely to be disease causing. So yeah, there's definitely a clinical element that goes into it.
Thank you.
Yeah, and I would just say it seems like there's going to be much more of a need on the gain of function or the epilepsy, early epileptic encephalopathy side for a variety of variants. Whereas if you're focusing on the loss of function side, you can get by with just a few, like the ones that Jeff Pitt just developed, that Yan Yang developed, the old Mauricio Montal heterozygote, and Jennifer just made one by accident. Those actually help us with a large number. It's a large family of variants can be modeled fairly well with that group. It's when you get into different ways of generating seizures that things get complicated, and it does make sense to pick and choose there carefully.
I have a question. It might have not, maybe it's not something that you can answer now, but I'm curious to know, like, before you get into clinical trials with humans, is there a number specifically of mouse or mice that you have to test and just, safely rescue them before you can actually go to humans?
Yeah, so I think there's no set number. What the FDA wants to see is that you had an experiment that was large enough and had enough statistical power to answer the question. So that's on the efficacy side. And then on the safety side, there's a whole nother set of animal studies that have to be done. Those are actually quite scripted, what exactly needs to be done. And that's usually in 2 species as well. So it would be, often it's a rodent species, and then dog is often the second species. But, so there's for a novel compound, there's a fair amount of animal work that goes into it before you can move forward. If it's something that's already FDA approved for another indication, so that's the repurposed drugs, it can be moved fairly quickly. If it's approved for adults, you may have to do some safety studies in juvenile animals, so some juvenile tox studies. But usually that's just in a rodent, and then you can move forward. from there. So it really depends on what the compound is. But, you know, we just published something with a Dravet model, with a novel compound that I think our group sizes to get the appropriate power were 50 animals per group. And that's, again, because the seizure frequency in these genetic models is relatively low. So you have to have a lot of animals, but that's what you need to do. And a lot of the failures that sort of the famous failures from particularly neurology, but also neuropsychiatry, when you go back and you look at the studies, they have group sizes of like 5 or 6. And they would definitely not pass muster these days. So, you know, I think there has to be a statistical rationale for the number of animals, but it's not really a set number.
Thanks.
On that same note, so as you both know, in SCN2A, a lot of kids don't have seizures. We have a very large part of our loss of function families that don't have any seizures. And we're hoping to be able to prove that drugs work in these children as well. And then there's some gain of function children that have stopped having seizures. Maybe they're on a drug that's working. How can we use the mice to look for other biomarkers that we can convince the industry groups that are worth looking at? Just behavioral stuff, or is there anything else besides seizures that they can look at?
Definitely. These are the exact questions that we're trying to tackle in the lab, and you ended up funding through the Action Potential grants one of the big studies in our lab to try and answer one of those questions. So we're very grateful of that. I didn't want to talk about it today because I didn't want to take away from other random animals, but one of the things that SCN2A is very, very important for is a part of your brain. We've been studying neocortex, both of our labs, in terms of seizures and higher order cognition, but there's another part under here called the cerebellum that's very important in motor learning, coordination, actually a lot of aspects of social interaction and social learning, where we've identified a very robust effect in the loss of function case in terms of your ability to hold a visual scene stable when your head moves. And this actually pops up very clearly in the mice. And we're in the process of repeatedly failing to properly write an institutional review board protocol or an IRB for human studies, because I have no idea what I'm doing with an IRB. And every time I write it, I do something wrong. So I have a very, very kind pre-review analyst that is helping guide me through this. But the goal is to actually see whether or not some of the kiddos have a similar phenotype. And so we're going to build a little helmet and a little eye tracker and let them explore the world and see if we can try and correlate their head movements with their eye movements and see whether or not there's any alteration. It's actually, what's remarkable to us is that one way of thinking about it is that the SCN2A loss of function kiddos, the mice are actually better at it than a neurotypical mouse. And that's the real question, is whether or not the kiddos are also better at this sort of thing. And whether that could be a biomarker. Because at the end of the day, whether or not they're better or worse, it's a little bit different. And that difference can actually help us understand whether or not, independent of seizure, whether or not the kids are responding properly to therapeutic. So that's one of the things we're trying to do. And actually, one of the exclusion criteria I think I have to set up is that the kids can't have a seizure to be in the first of these studies. And so we'll try and explore that as best we can in the coming months as we build this crazy little device and hopefully pass the IRB board.
That is so cool. Thank you for explaining that. And I just want to clarify, like a biomarker means that you test them
Mhm.
and you get a result, and then you give them a treatment, not you personally, but they go through a clinical trial, have a treatment, and then they take that same test again, and you're looking for a change after they've had the treatment.
Exactly, exactly. So biomarkers are just assays that allow you to tell whether or not your child is neurotypical or not, and then whether or not any sort of therapeutic you're trying to use has any efficacy. And this, as I've learned more and more in the field, with epilepsy, we have an advantage, if you permit the crude way of speaking, because you can quantify that. You can do an EEG, you can record the number of seizures, you can record the way the seizures look, and you can see whether or not their frequency and severity changes. With something like intellectual disability, developmental delay, autism spectrum disorder, many of the assays that have been used in the field are a little fuzzier, a little more qualitative. So for example, you have a score sheet and you say, how social is this child? How much do they play with this particular toy? And on a rank of 1 to 4. And this is how many of the drugs that have been tested in the autism field have been assayed. And the thing about it is that with all these clinical trials, you have to decide what's called a primary endpoint is before you even start. And so you better guess right. And when the analysis is fuzzy just off the bat, then it's really hard to identify or to even choose what your primary endpoint should be. And so there are very famous stories of some of the best drugs ever that worked beautifully in a mouse to rescue social deficits in mice, failed their primary endpoints when tested in human, despite some people saying, well, for my kid, it actually works pretty well on these other things that you weren't even asking about, that you weren't even testing for. And so coming up with something that is a little more quantitative and relies more on physiology that we understand extremely well, and in some ways is purely reflexive physiology, is something that I think could be useful, especially for SCN2A.
Absolutely. We spoke with Caitlin Hudak recently. She did a table talk, and she also explained about her EEG, and that could be used as a potential biomarker as well. So
exactly, they've identified a beautiful auditory response that is very clearly different, and so that would be another great assay to use.
These are so important and so critical, and also a lot of the reason why we're doing the CTRS, which is looking at the measurements to make sure that they're the right measurements before we go into clinical trials, because we want the trials to be successful. Thanks for explaining that. Jennifer, I have a very silly question. Why is the A lowercase when we talk about mice, but it's capitalized when we talk about the gene?
So
in humans,
yeah, no, nomenclature is fun. So human genes are all capital letters and italicized for the gene, and depending on who you talk to, the protein is all capitalized, not italicized. So that's humans. For mice, it's capital F and then lowercase SCN2A and italicized. So that's the mouse gene. In mice, again, some people would say you can use all capital SCN2A to indicate protein. But we all have this other nomenclature of NAV 1.2 that doesn't have any species indication associated with it. So yeah, there are all kinds of rules. There are committees that spend all their time thinking about these rules and correcting when people don't get them quite right. And so
I think it was Dr. Miriam Meisler who, when we first started the foundation, we had a lowercase a, and she
Yep.
She told us that is wrong. Do not
Yes.
change everything. Thank goodness.
Yes, and I that is where I learned my nomenclature. So
That's great. Does anyone else have any questions? Feel free. This is your opportunity to ask questions to the professionals. I have a question. Are deletions always a loss of function in SCN2A?
I would say
good
yes.
question. Yeah, so I would say yes, but if they're in the last exon, so the last exon is actually quite large, and the way that things usually work, if you have a truncation early in a gene, it will get translated into mRNA, and the mRNA gets degraded before it ever makes protein. So that's nonsense mediated decay, and that's a quality control checkpoint in cells. However, if there's a deletion in the last exon, it's not picked up by this quality control system, so you can get protein. And if you're missing part, and it largely includes the C terminus, if you're missing some of those important signaling sites in the C terminus that hangs in the cytoplasm and interacts with all kinds of things in the cell, you could have some potential, I don't know if there would be gain of function, but certainly change of function effects.
Yeah,
from a function, it would still be a functional channel, but it wouldn't function appropriately.
Yeah. And I don't know whether it would traffic properly. In many ways, what Jeff Pitt's lab made with the 1903 should have been one of those channels, but it actually looks like it behaves like a nonsense mediated decay.
Yeah, maybe. I don't know. The problem is the antibodies are all,
Also
or the antibodies that work are all way upstream, or not way upstream, sorry, way downstream. So you actually, and we're running up against this with our Frameshift as well, like if you use an antibody that detects an epitope that's downstream of your deletion, so you're not even expecting that to be there, of course you get a blank Western blot because there's no epitope, but it doesn't mean there's no protein. So you have to kind of choose your antibody wisely. And we see that all the time, that people don't think about that. And it's often the epitope isn't present. I don't think that's the case for his, but.
That helps to understand the deletions a little bit, but just recently, actually on our last table talk, we talked about a variant that is a protein truncating variant. So it's TYR84X stop, and that one actually carries a current. It's been found to carry a current. What does that mean? Does that mean it has a little bit of a gain of function? Is it still a loss of function?
So that must pick up a different start codon, because that's super early.
Yeah.
That's super early in the protein. So there must be an alternative start site that it's picking up. That's interesting. I don't know.
I sent it to Al.
Yeah. I think we need to know more about it. That would be a good iPSC cell, some looking at transcripts and protein. That would actually, because in a heterologous system, you might make a cDNA, and it might not do the same thing because it's not being processed in the same way that pre-mRNA and mRNA are processed in a neuron.
Sorry, I got off the mouse model thing. I just had those two quick questions that I wanted to make sure I got answered for the families. Thank you so much. Catalina, I'll turn it back over to you. Sorry. You're on mute.
Catalina, yeah, muted.
Oh my goodness. Okay, I'm so sorry. I was saying that I wanted to thank you again for that amazing session. I think we have all learned a lot after today. It was quite exciting to understand more about how things work backstage and how much it's involved in finding something for our kids. We truly appreciate all the work you're doing. And thank you to the families that have joined us today. All these questions, I hope that the answers were insightful. And I know that most of the questions will come to our minds once we're off the talk. So hopefully we can send those questions later on through Leah or through any of the board members. And just to reassure the families that we are here for support and for anything that they might need. We have another 2 talks left, and I hope we can all register and participate. And we will be making this recording also available soon. And just a reminder that, there's always so much work to do, and that we can all participate by, just getting involved in the SCN2A clinical trial readiness study. If you haven't registered, please go and register. And then also we're having the SCN2A World Challenge, the virtual walk. Register your team and do that work for our kids. And well, that will be it. And those links for registering to our walk and to the SCN2A clinical trial readiness study will be in the website and at the end of this recording too. Thank you so much.
Thanks for having us.
Thanks.
Thank you.
Thank you.